I want to thank the Illinois Mycological Association for inviting me to do this program. It's a hybrid meeting up at our Niles location and on Zoom. So welcome to the our crowd of 40 people in the room and also everybody on Zoom. This program I've given quite more than 10 years ago for some different workshops and the club and so forth. And it's also a talk I do for a workshop or one-day class out at the Morton Arboretum that's every 2 or 3 years. So this is about how to look at mushrooms more carefully. And then at the end of this talk, we're going to sort different kinds of mushrooms into groups based on what is called stature types. So it's like a combination of characters to put them into groups, and that helps you narrow down the mushroom identity to fewer options. So there's lots of different mushrooms out there. There's edible mushrooms in a lot of different of these groups. And tonight we're looking mostly at gilled mushrooms and their characters, but this would also apply to boletes, and some of the characters would apply to other types of mushrooms. Basically the different surface features and different colors and so forth. But each type of mushroom fruit body or macrofungus fruit body has its own kinds of terms for some of the parts. So tonight for gilled mushrooms, these are the main characters for mushroom ID. For gilled mushrooms and also boletes, spore print color helps. Cap features, and then what's under the cap, if that's gills or pores or teeth, and those features. The stem and the features on the stem, the veils under the cap or on the stem, we'll look at those. And then things I'm not going to say much about would be odor and taste. We had a foray last Saturday with lots of different odors, good and bad, which was fun. And chemical reactions would be something like with potassium hydroxide or ammonia reagents. There's some groups like boletes and stuff that's useful. Habit is how you find the mushrooms, whether they're solitary or grouped or in a larger. We had some troops of tricholoma and stuff yesterday, where you have a whole bunch of, like a little army of them on the ground. Ecology and habitat are quite important. That's a whole nother topic on where you find the mushrooms, what the trees are, what the kind of habitat. For example, this weekend, we were at woodlands, so oaks were our main mycorrhizal host for a lot of the mushrooms we found. And then chemistry would be a more advanced stuff you do in a lab, and we don't do much of that with mushrooms. And then later you can use microscopic characters. And then we have some people in the club that are now saving specimens and doing DNA sequencing with those to pin down the identities of those. So we're going to focus on some of the top upper list here on traits for the mushrooms. Spore print colors are all over not quite all over the map. There's lots of different colors, mostly in these 4 categories that your book would use: white, pinkish, different shades of brown, and then dark spores. And if you're starting out, you might not realize that the white category includes a whole bunch of pale colors, not just white. A lot of mushrooms have white spores, but there's also different colors of buff and cream and pale yellow. There's a few that are pale lilac, a few that are pale green, and a few that are a light pink, more like a lipstick pink than a brownish pink. And then the standard pinkish category includes salmon and brownish pink. And there's a couple, mainly 2 families that have that color. There's all sorts of brown colored spores, from lighter brown, ochre, rust brown, earth or clay brown, it's like a yellow brown or an orange brown. And then under dark spores, there's a couple families with that, and those are chocolate brown, like with the store mushroom agaricus, and purplish brown or black. We have black spores in like the inky caps. The cap is the top part of a mushroom, and fancy word for that is pileus. You can think of pileated woodpecker. It's got a crest on the top. So that's where that word comes from, meaning a cap. So cap characters would include the shape of the cap. What the margin is doing, that's the edge of the cap. Does it have extra features on the margin? Is the margin curved down like in this drawing, or is it curved up? And are there other things like ornamentation on the margin? Dimensions is the size of the cap, how tall it is, and usually more importantly, how broad or wide, or what's the diameter of the cap. And then I split the surface features into 3 different categories. Topography is like the physical structure of the cap. Like, does it have scales, or does it have zones and stuff like that? And then the vestiture is things like scales and warts and hairs. And then wetness is if it's dry or slimy. And then there's a couple different kinds of veils, and they leave pieces behind on the cap, the stem, to tell us that it had a veil when it was younger. And so that's a feature to look for when you're out collecting mushrooms, is get a younger mushroom, find younger mushrooms, and they would show different stages of the cap opening up. So you might see the young veil on the younger examples. The context or flesh is the inside of the cap and the stem, and that has different textures and colors. And then the mushroom itself would have textures, and then there's colors everywhere. And under this category, we're talking about the cap. So what is the color of the cap? Does the cap stain when you bruise it, or does it have reactions with chemicals? The stem is going to have a lot of the same characters, the shape of the stem, what's going on with the base. That's really important. So when you collect a mushroom, you want to pull up or dig out the base so you have the entire stem, because some things like amanitas and other things have particular things going on at the base of the stem that might be partly underground. And then how long the stem is and how wide the stem. And then again, it's all those surface features and the veils, the context, texture, and then the color and the reactions to the stem. The stem is under the cap and holds up the cap above the ground, so that when the spores are made under the cap, they can be liberated and get out into the air. The gills are underneath the cap for a gilled mushroom, and those have their own characters. Attachment, we'll look at that. Spacing, how crowded they are. If there's lamellulae, which isn't that important, but it's something you can look for. Those are short gills that don't go all the way to the stem. The margin of the gills might be different. Breadth is how deep the gills are, if they're wide or broad, or if they're narrow gills. And then if you want, you can measure the length and breadth of the gills, but that's mainly dependent on the cap size. And then gills have also to have different textures and different colors and staining. So first we'll look at gill attachments. These drawings are from Wikipedia. So first we have free gills, and those are gills that are attached to the cap, but they don't go over and attach to the stem directly. There might be either a bigger gap or a smaller gap next to the stem. Succeeding gills are gills that are attached to the stem, but they can split away as the cap expands and pulls the gills up. So there's a few things that have succeeding gills. So that might make it a little tricky, but sometimes you can usually see that that's what's happening. And you can see the attachment points on the stem after they break off. So here's 2 examples of free gills. We have a Lepiota on the left with white spores, and the gills there curve up into the cap instead of going over to the stem. And then pluteus, that has pinkish brown spores there on the left, on the right, lower right, that has a big gap between the gills and the stem. So in pluteus, with the big gap, we can call those remote. That means there's a big distance between the cap and between the gills and the stem there. One character often associated with free gills that had more attention about 100 years ago was that Some of these things like amanita, lepiota with free gills often have a difference in texture or continuity between the cap and the stem. And you can kind of wobble the cap and detach it from the stem like a ball and socket joint. Whereas mushrooms with attached gills usually can't cleanly break the cap off of the stem like that. There's different kinds of attached gills. These reach over and join the stem. Adnate is a common condition where they are broadly attached to the stem. And then adnext, they're more narrowly attached. And this is sort of a gradation between adnate and adnext. But basically they're attached. Usually for most mushroom IDs, it doesn't matter if they're adnate or adnext, just that they're attached. And then there's examples of that. So on the left, we have a Lacaria ochre purpurea with really broadly attached, very broad gills and broad attachment on that stem. And on the right, we have a different Lacaria where the gills are adnext. They go up into the top of the stem there. And then the last gill attachment type is decurrent, and that's where the attachment runs down the stem. And that's a defining feature of the Clitosobi group, which have these sloping gills. Subdecurrent is the same as short decurrent. That's where the gills go down the stem a little ways. Here's an example of short decurrent with this Lactarius on the left, and then a drawing of a Clitosobi type thing with a funnel-shaped cap that gets uplifted and accentuates that decurrent gills. The decurrent gills running down the stem. Then I forgot notch gills. So that is a feature where the gill comes across and then there's a kind of a gap or a cutout before it reaches the stem. And there's technically two kinds of notch gills. And your book probably usually doesn't distinguish between the two. But sinuate, like the letter S, the gills go up and then down on the stem. So they're a little bit decurrent on the stem with that notch. And then emarginate goes up and then over into the stem. So it's more like a notch with the adnext connection there. So on the top, we've got marginate example. And then lower down there, we've got sinuate ones where you can see this in a cross section, or for sinuate, sorry, like this, you can kind of look down under the cap and see how the gills bend around and connect to the stem. Gill spacing is looking at if the gills are crowded or close or distant. It's how they're spaced, how much space is between the gills. So mushrooms differ in that regard. Sorry. Forked gills, that's pretty uncommon. There's a couple of distinctive things with forked gills like Cantharellula and Hygrophilopsis, where the gill comes up from the stem and then forks repeatedly. You can call this dichotomous forking. There's a regular branching of the gills there on the left example there between the stem and going up to the cap. This is a way to fill in that space with more gills, but they're attached instead of separate long gills and short gills. And then a few mushrooms like Lactarius have a latex in the tissue, which is most easily seen by cutting the gills and having that latex come out. And that would be another feature where different Lactarius or milk caps have different colored latex, and then the latex may stain a particular color like green or purple or something, or change to yellow. Another gill feature you might see is in Lentinellus and some things like that, where the edge of the gill is broken up and it's called serrate. Or if it's really irregular, you can call it eroded. So Lentinellus and Lentinus are the typical members that have that. Marginate gills have a something different on the edge, usually a different color like this. So we have Bacterius lignotus, the chocolate milky, and that has little microscopic cells called cystidia, and that pigment in those cells is on the cap and on the stem, but also on the edges of the gill. So you get this little brown edge to the gill, which is really pretty. And then mycena leyana, which is common, has red cystidia on the edge of the gill. And then this other mycena, Purpurea fusca, has more of a purple color in those cells on the edge of the gill. And those are all white spored mushrooms. If you have a dark spored mushroom like a Satharella or an ossivy, sometimes you get the cystidia, which are the little sterile cells. They will reflect the light if they're kind of present and spherical or whatever, have a good shape. And they reflect the light and they look like little tiny crystals on the edge of the cap. So that's called, I call it white fimbriae. It might have other words, but that's something to look for with a brown spored mushroom, is whether the edges of the gills have this little kind of white crystal looking. And this is not all across the species. So like an ossivy, some species would have this and some don't. So there's lots of different features for caps and stems. This drawing is kind of a hybrid by Gary Linkoff's book, with sort of a hybrid mushroom with lots of different features on it. We're going to look at veils, and this slide's kind of technical, but it shows the 4 basic kinds of veil combinations you can get. There's universal and partial veil. So universal veil, like in an amanita, covers the entire mushroom when it's in the button stage. And a partial veil just goes under the cap. It joins the cap to the stem. So we have a case here where we have a universal veil in the top left, and this is Vulvariella or vulva pluteus types, where it's got a big vulva, but no ring on the stem. And most amanita, pretty much all the amanitas have some sort of universal veil. There's technical words under there that nobody uses, but you can kind of figure out what they mean. So these 4 mushrooms all have veils. So they are all velangiocarpic. That means, if you can see on the bottom there, veil enclosed fruit. But then the veils are different. So the universal veil, we just have the universal veil. So that's mono velangiocarpic. And then on the right top, we've got both veils. So that's bi-velangiocarpic. But basically, you don't need to know that. You just need to know it's got a universal veil and a partial veil. A lot of amanitas have that character, like that Amanita mysterious there. And then agaricus and stuff, other stuff, Lepiota, whatever, have a partial veil under the cap. And that's paravelangiocarpic. And then there's an interesting case where you can argue that on the bottom right there, you've got a foliota type or a leucofoliota type thing there. It's actually a leucofoliota decorosa. It's really pretty mushroom. And you can argue that that has a union of a veil or a weird veil that is fused to the cap and the lower stem, but leaves a texture-free zone above the stem, below where the veil is connected to the stem. So when that mushroom opens up, that kind of surface features on the cap and the stem break it where the veil is, and then the upper stem looks completely different, usually a different color and more less ornamented. So that's a meta-velangiocarpic type. So you can look for these different kinds of veils. A lot of mushrooms don't have any kind of veil at all. So here's an example. Pluteus on the left there doesn't have any veils, but it's got free gills and pink spores. And then Vulvariella is in the same family. It's got free gills and pink spores, but it has a vulva. So it has a universal veil that breaks and leaves that cup at the base. And little Vulvariellas that you find in the woods, you may not recognize it as Vulvariella. So when you pick these things up, again, it's important to get the base to make sure you have that vulva. Otherwise, you're going to lose that and think it's a weird Pluteus on the ground. Here's Amanita. They often have both veils. And this is a drawing I did I don't know, 30 years ago sometime, when I was starting out, showing Amanita button opening up and the vulva breaking away and leaving a patch on the cap, and then the partial veil under the cap breaking and leaving a ring on the stem there. So if you get Britt's Amanita book, you'll see all the different Amanitas we have around the country, and nice photos of those. Here's some examples of different kinds of veils in Amanita. So on the left, we've got the Amanita Jacksonia, or the Caesar type mushroom thing, with a white universal veil that leaves a really big cup at the base. And the bottom left there, you can see the little button where if you're collecting puffballs, you want to cut them open and make sure you don't have a baby Amanita inside there. So then that white vulva veil breaks and the stipe expands. The cap gets lifted up, and then the cap expands, and that partial veil breaks and leaves that nice skirt or ring on the stem. So these are both very clean veils that are membranous and leave definite nice structures. The Amanita muscaria on the right is a bit more messy with the veil. It's a weaker veil material. So when the cap expands, that veil material kind of breaks up into ridges on the stem and onto separate pieces or warts on the cap. And then the partial veil breaks and leaves a ring on the stem. But in Amanita, there's a whole range of these gradations between a very firm membranous veil, like on the left, over to in the center, we got weaker veils that are leaving different sized warts and patches on the cap. And then on the right, we've got a powdery veil that leaves little powdery warts or material on the cap. And then down below, you can see different types of what happens on the base of the stem with the different kinds of veils, going from a cup type thing to circumcisal, which is like a booted ring or like a sock around the base, and then a deeper cup there, and then circumcisal is where the veil breaks up into rings. And then some of these Amanitas with cottony, fluffy veils, they leave a whole lot of little cottony debris on the stem. Here's some other rings and other types of mushrooms. We've got top left, that's an Agaricus bitorquis, which is bitorquis refers to the double ring. It's got one partial veil, but the way the cap joins to the stem, you get a fake or false ring on the bottom there. That kind of curved band is where the cap was pushing against the stem. And then we got a Suillus luteus there in the lower right, lower left, with a baggy veil, which is kind of unusual. And then we've got another Amanita up there with a skirt, and then a Lepiota type parasol mushroom with a ring there. And rings after they break can go up or hang down, or have striations. They often have different colors. And then cortina is a cobwebby veil made up of fibers. And we have that in Cortinarius. So a cortina is a cobwebby veil that gives Cortinarius its name. And that is a partial veil made of hairs or hyphal strands. And when that breaks, the hairs hang down on the stem and often trap the rusty brown spores. You end up with a zone of hairs, and they end up orange from the spores and getting stuck on there. Some veils are really weak and don't leave any material on the stem, but they'll leave little patches hanging on the edge of the cap, like in some of the sapphirellas, and a few other little brown mushrooms like tuberia have even more obscure veil remnants on the edge of the cap. There's lots of different shapes of caps. You can look online or in your book that you might have to see which words they use for the different shapes. Some of the basic shapes are cone shape. Convex is like rounded. An umbonate cap has a bump in the center, and it can be a big bump or a small bump. And then campanulate refers to a bell shape. And then some of the caps will change shape as they open up. So you can have a young mushroom that's convex And like an amanita, it opens up and is flat. And then there's other things, plutosopy and similar things that are funnel shaped. Some lactarius and stuff are funnel shaped, where the cap opens up and expands up. It's kind of folded up as it grows, and you get this depression in the center. There's some little mushrooms that are umbilicate like a belly button and have a little depression in the center of the cap. So now we have the surface features here. And I have not found better words than what I call topography and vestiture. So topography is like more like the landscape, physical features of the actual cap surface. If there's ridges or bumps or grooves and so forth. And the word for, if you want to be most clear, a surface that is even means it doesn't have any bumps or ridges or grooves. And for vestiture, that's hairs and scales and warts and all that other stuff. And a glabrous surface doesn't have any of those. But a lot of books and descriptions will use the word smooth. And that's usually a combination of even and glabrous, because that's sort of the default condition of caps not having any features on them. But there are quite a range of features on caps, patches and fibrils and scales. And then these would be different colors and sizes and shapes and all that. And also you can have zonate features on some of this stuff. So one thing that I like to keep clear is scales versus warts. There's a few people I know that don't make a distinction, but there's the difference is whether these structures are actually attached and part of the cap surface. So lepiotas and foliota and things like that, they have scales that are like kind of like shingles and they stick out on the top of the cap. And then warts are, or patches are from amanitas, things that have a universal veil that covers the cap, and then part of that veil material gets left behind on top of the cap. And as the cap expands, it gets broken up into warts, like on the right there. So here's scales. So scales are part of the cap surface. And what happens here is that the outside of the cap dries out because it's exposed to air, and the hyphae on the outside are not keeping up with their extending length to keep up with the expanding cap on the underside of that top layer. So the outer layer of hyphae break up and make those scales, because the underparts are expanding faster than the scales on. And then warts are patches of veil that are just sitting on top and left behind when that veil breaks. So these, you can rub them off or the rain can wash them off. They're not part of the actual cap surface itself. And then scales are different sizes and colors and so forth. They can stick up or be flat, all sorts of variations on scales. Surface wetness ranges from dry. Most mushrooms are kind of on the dry or moist side, where they feel damp maybe. And then if they have some sort of viscid layer, it might be tacky or sticky. And if you're wondering if a cap is tacky or sticky, you can moisten your finger and then push it on the cap and see if it's kind of sticky. Because when the caps dry out, they can start to lose that. And then these examples here, the parrot mushroom lower left has a viscid cap. It's got a thicker layer of slime there. And then that Cortinarius on the right has a really viscid or glutinous slimy layer that can actually drip off. One idea for why some mushrooms have this is to keep rainwater from soaking into the cap, so the cap doesn't get too soggy. Limacella very often is a relative of Amanita, and had, instead of a membrane universal veil, it's got a more slimy universal veil that leaves a slimy cap and usually a slimy stem. And we've got 2 or 3 species of those around the Midwest here. So that's a neat thing to look for. This is a character you can probably see in your lawn if you have little lawn mushrooms that come up. Hygrophanous refers to the water or the moisture in the cap. And that is a color change when the cap dries out. So when the cap is moist, it's going to be a darker brown or darker color. And that's from the translucent cap surface. So you're actually kind of getting some color from the inner parts of the cap. Often the gill color is kind of showing through the cap surface. And then when that cap loses moisture, it becomes opaque. And then it reflects the light. So you get a pale spots on the cap. So these caps change color as they dry out. Here's a couple margin features. Striate margin is a physical feature of the cap. So there's actually ridges or grooves in the edge of the cap, like on the left there. There's like an amanita and maybe a mycena or something there. Similar to that is translucent striate, but in this case, you see lines on the edge of the cap, but actually what you're doing is you're looking through sort of the translucent cap and seeing the gill attachments underneath. So you're looking through and seeing the lines of where the gills are. If you actually look carefully at the cap surface, there's no lines or grooves on the cap. It's a visual appearance of seeing the gills attached underneath. Two other examples. We've got grooved caps in this little inky cap. So we got nice pleated or gilled grooved caps. And we also have little frosted granules on here. So we got two different features. We have a physical feature of the grooves, and then we got an ornamentation with those white crystals type of stuff on there. One of my favorite Lactarius is on the right, Lactarius torminosus. It has a zonate cap on the top, and then the margin underneath has a beard. So it's got lots of these long hairs or fibers that come out and the edges rolled up, which helps protect the gills when the cap's younger. Stems, lots of different stem shapes. And your book would have different names for those, or your website, whatever you're looking at. Some of the basic ones, equal means the stem is the same diameter from the top to the bottom of the stem. Club shaped has an enlarged base. Bulbous is where the base is enlarged abruptly, more like kind of an onion. And then amanitas and things have a cup. And then there's a few mushrooms we have with a rooting stem, where the stem comes up from deeper in the ground and gets bigger as it comes up to the surface. So the thing I always point out is these rooting stems grow up. They don't grow down like a carrot. They're growing up from beneath and getting bigger as they reach the soil surface. A few mushrooms have rhizoids or cords at the base, which is something you can look for. Most mushrooms have a central stem attachment where the cap is evenly centered on the stem. There's a few things that have an eccentric or off-center stem, and then things like oyster mushrooms and similar ones have a lateral stem that's attached to the side. There's all sorts of stem surfaces, just like cap surfaces, and different words for those. Another thing you can look at, especially for smaller mushrooms, is what's going on inside the stem. Is it solid? Is it stuffed? Which looks like it's kind of a looser texture inside, like it's got cotton stuffing. If it's hollow, it might be tubular, which is basically hollow. I don't know why. I don't know the difference between tubular and hollow there. Fistulose is sort of like a narrow hollow type. And then that russula has a chambered stem, which you can also call lacunose if you want. So that's kind of a fun, and that's russulas have a different texture. So that's a different way that the stem expands and opens up with these chambers. Now we're going to look at some different examples and see what the features are they have. So on the left, we've got a Hemistropharia albocrenulata. And that is a brown spored mushroom. So it's got mature gills here are kind of brown, dark brown. Albocrenulata refers to the white edge on the gills, which don't show up as well here. But it's like that other photo I showed where it's got the white crystals on the edge of the gills. This has a viscid cap. It's sticky cap, and it's got scales. Most of the foliotas have scales, but one thing with foliotas is some foliota caps are dry and some are viscid or slimy or sticky. And the one thing to look at with a foliota with sticky caps is whether the scales are dry or sticky, because there's some variation there too. And then on the right, we've got a panis. We basically have two panis around here, one that's hairy and one that's not. And this is Panis rudis or Panis neostrigosus with kind of purpley color in the spring, and then it turns brown. But it's got a very hairy, sort of hairy, scaly cap, and it's funnel shaped, which gives it decurrent gills, and then the stem is also fuzzy like the cap. So it's got a nice texture in there, and white spores. Wow. Here's an example of two fungi that would have been Clitospi ages and ages ago because they have decurrent gills. And we have several of these little funnel-shaped brown mushrooms around, and you kind of have to look carefully to see what the features are to figure out which is which. And here's two of those examples. And some of these little mushrooms have the biggest names. This is on the left, we've got pseudo-armillariella, because it kind of, somebody thought it looked like armillaria back when armillaria was called armillariella, because armillaria was split between tricholoma and armillariella for a while. But this one on the left has radial hairs, which are very small. In that inset photo, you can kind of see the hairs on there. So the cap surface kind of looks streaked, but with a hand lens, you can see those streaks are made up of dark hairs. The one on the right, the pseudoclitospi, which looked like a clitospi, cyathiformis, which means it's shaped like a goblet. It's got a funnel shape, and it has lines on the cap, but that's translucent striate. So that doesn't have any physical lines on the cap, but you're looking through the cap surface at the gill attachments. These are both white spored on wood. Another similar one is Arenia, which is sort of similar to the pseudoclitospi. Here's one example of staining reactions. So there's a group of russulas called blackening russulas, and they stain, eventually stain dark or blackish. There's some other russulas that stain other colors. Russula densifolia stains red and then it turns black. So to take this picture, I scratched it, that lower scratch, and waited until that scratch was red, and then made that other scratch and waited until that scratch was red, and then took the picture. After the picture, that red scratch turned black. So it's just a sort of a time delay on the color reaction. Here's one of my favorite mushrooms from up north, Wisconsin. And I was looking this up today, and back more than 10 years ago, I thought this was Cortinarius torvus, but it doesn't seem to match online pictures of what people are calling Cortinarius torvus. But it's got some distinctive features of really broad, widely spaced gills and a ring. So it's one of the Cortinarius, sort of like the gypsy mushroom. It used to be Rosites, which is now Cortinarius capparatus, that has an actual membranous ring on it. So there's a few other Cortinarius that do have a ring like that. Here's another picture of that. And in a cross section, you can see how broad the gills are there. Here's another mushroom from up north. This is a more interesting inospe because it's got blue staining in the base. And some of you may know what that might mean. Some psilocybin or psilocybin type content in there. But this is a brown anospe. Most anospes are brown or golden brown. And this one has sort of scaly, hairy scales on the top and a scaly, hairy stem also. This also has those pale gill edges. And then this one is another northern mushroom. This is Gomphidius, and they are a weird group. They're more as conifers, so we don't see them around Chicago. But you wouldn't know looking at this, but this actually makes black spores. If you do the spore print, it has black spores, very dark spores. And it's a slimy mushroom. It's got a slimy cap. And that younger one there, you can see that white material joining the cap and the stem. That's a slime veil. So it's sort of a fibrillose veil that's slimy. So the cap and that veil and the lower stem is slimy. And then when that veil breaks, it leaves a little bit of a ring zone at the top there. And you can see that the ring zone is dark. That's the black spores getting trapped there. And then the long stem goes down in the moss and has sort of a scaly pattern. And then on these Gomphidius, what you need to do is make sure you pull up and get the base of the stem, because the base of the stem can be a different color, like this is yellow. So that's the pictures of mushrooms. And then we're going to look at using some of these features to put different mushroom genera into groups. And one of the references I used for this talk and for the handouts is David Largent's book on how to identify mushrooms of genus number 1, the one on the top left. And these you can find online. I have a search saved on eBay for when they show up and they're selling at a decent price. That sometimes they're overpriced online, so you want to shop around if you want to get these. But these are quite old. You can see they're 1977. If you're going to get, so for macroscopic features, the volume 1 is really worthwhile. It's got all these different macroscopic features explained and diagrammed with examples, black and white drawings and so forth. And what you want to do is get the second edition, which is more than, it's like double or triple number of pages. So the second edition is 1986. So when you're looking online, you want to check the page length. Make sure it's the one with the 166 pages and not the one with 80 pages or whatever it is. And that the longer book has some very useful keys and stuff in the second half of the book. Volume 2, so volume 1 I would recommend because that's macroscopic features. The next one I would recommend is microscopic features. So that's similar, but it's all the features you can see under the microscope, like all the different cell types and the mushroom cap, stem, and gills, and then all the different characters with the spores and everything. Number 2, you don't need that. That's field identification of genera. It's a very small book, and this is like Frisian genera, so it's old genus concepts. The number 4 there is also a small book. Number 5 is on cultural and development features, which, unless you're growing mushrooms or doing lab studies, probably not interested. The other one you might find interesting, even though it's from 1988, is number 6, Modern Genera. That's 277 pages, so that's a bigger book. And that goes through the modern genera that came out of these older genus concepts. So like, so when Linnaeus started naming things, he had like agaricus and amanita and just, a dozen or so mushroom names. And then Fries and his colleagues at that time made a lot of different genera, agaricus and clitospe and all these things. And then our modern genera are kind of split out of those older groups. So in this volume 6, you would go and there would be a page for like clitospe, and it describes that group and all the modern genera at that time that were split out of clitospe or are like clitospe with the decurrent gills. So it's a useful book to figure out concepts for different genera, but because it's more than 20 years old, it's a little bit out of date, but still would be useful if you want to learn different genus concepts for gilled mushrooms. So if you go to my website, mycoguide.com, you can go to methods, and I have handouts on there that I use for classes. And the top section is for identification, and the gilled mushroom stature types is what I'm going to show you here. And that is a 2-page handout. And that splits gilled mushrooms into different stature groups that are in Largen's book. And these are based on the veils and the gill attachment. And then the second page brings in spore color to split the groups up. So we'll kind of run through this. So here's the top part of page 1. So stature types. So on the top row, we've got all the gilled mushrooms stature types with free gills. So those cross-section drawings, you can see all those gills are free, so all those caps are the same. The difference is what the veils are doing. So an amanitoid, the amanita type stature has a partial veil and universal veil. It's got a cup or some structure, and then a ring on the stem. Vaginite type amanitas, which would include Volvariella, has free gills but a vulva, but there's no ring. And then Lepiota type has a ring but no universal veil. And then the pluteus type has free gills, but there's no veils. So there's 4 different free gill types there, and the differences are the veils. The next row is attached gills. Those all have attached gills, and these are all mostly larger, more fleshy mushrooms with fibrous or fleshy stems. They're not little skinny stem mushrooms. The first one is the one with the partial veil, and that's an armillaria type. So it's got an annulus, but it's got any type of gill attachment. So anything with a ring that doesn't have free gills goes in that category. And then the tricholoma type has notched gills. So sinuate or maybe emarginate goes in that group. And then the naucorioid has attached gills that are not notched. And the Clitospe group has decurrent gills. So these are split on, the first one has a veil, and the other 3 have different types of gill attachments. So Naucoria is an old genus name we really don't use much anymore. And then in the bottom part of the chart, on the bottom we have pleuritoid, where the stem is attached to the side or on the edge of the cap, or it doesn't have a stem at all, like some of the Crepidotus types. So the oyster mushrooms and little things like that are down in the bottom row. And then that third row there on the top of this picture is the little mushrooms with more cartilaginous stems. So the stem is maybe kind of fibrous, but it's more skinny. And it's a smaller mushroom, and some of these have cartilaginous stems, which means the stems are more rubbery and kind of flexible, kind of bendy like cartilage. Again, that first box, the Galerina box, those are anything with a ring. And then those other 3 are cap shapes. So mycena have the bell-shaped cap. Clivia have a rounded cap. And the Omphalina group have a dimple or umbilicus or belly button type center of the cap. So that's how those are split up. The back page has these stature types down the left column there. So you can see Amanita, Vaginatoid, Lepiota type, pleuritoid, Pluteus type, down to Clitospe type for the top of the page. And then across the top are the 4 categories of spore print pillars. So what you do is you figure out which stature type you have, and then with a spore print, you figure out what color spores you have, and then you can see the combination of the 2 tells you which box on this table to look in for possible genus names. So you can see some of the genus names like Amanita are repeated around the place because they all have white spores, but they have different aspects with the veils, different kinds of veils. And then the The pink spores, that second column, pink spores, you can see there's not too many things in there. There's little variella, bulba pluteus, and pluteus, and then entelomas and clutophilus, and some of the segregate genera under Amyloma. There's a whole bunch of brown spored stuff. That first column has tons of white spored stuff. And then the column on the right is all the dark spores. So you can see agaricus is over there along with the inky caps and other things. So I added, this handout was made quite a while ago. I added a few things on this slide, like Arenia, or the Clitospe type ones that, Arenia is pretty common around here. Pseudoarmillaria is not, you can find it up north. And then Gomphidius, I don't have on this chart because we don't find it in Chicago. So if you look at the top, it says Chicago region. So there'd be, if you're using this up in Wisconsin or Michigan or Minnesota or other places in the country or Canada, there would be a few other genera you could add in here for possibilities. On the bottom part, we've got the small mushrooms and then the Pleurotus group on the bottom. There's no pleuritoid group that I know of those black spores, but you can see there's lots of things in that page with white spores, brown spores, and some dark spores. So as an example, two examples here, that Cortinarius torvis would have attached gills that have a fleshy fibrous stem, and it has an annulus, so that stature would be the armillaria type or armillarioid type. And then with the rust brown spores, that would include Cortinarius under there. So if you're using the keys on Michael Quo's page or website, this is some of the characters that would be initial characters to help you figure out which group you're in, the spore print color especially, and then the cap and gill features and veils and all that stuff. And then Gomphidius up north has attached gills, fleshy stem. It more or less has an annulus with a viscid veil, but it has decurrent gills. So it's an armillaria or Clitospe type, depending on whether you recognize it as a veil or not, whether you notice that. And then the black spores would help you get it into Gomphidius or Chrogomphus type. But this is up north and other places. So that's it for the talk, and we can do some questions, live questions and Zoom questions. Any questions from the floor? So the question about the stature types, if they're still used or promoted or whatever, I don't see them used all that much, but they're sort of indirectly done that because of keys and books and websites will ask you, spore print color and veils and gill attachments, stuff like that. So it's sort of sorting you into a category, not that it's labeling that as a stature type necessarily.
We have a question from online.
Okay.
Are all Amanitas toxic poisonous?
Are all Amanitas toxic or poisonous? No. Some of them are deadly and some are good edibles. If you're living on the West Coast, especially California, you want to join the club so you know which are the good edible ones. We don't really eat any Amanitas around here. There might be a few clubs out east that have the Caesar type mushroom. We don't have that one here. We're too far north. So Amanita ranges all the way from really excellent edibles that were enjoyed by the Roman emperors, all the way to deadly mushrooms that'll kill you in 3 days. So also some other edible mushrooms, like your store mushroom, is an agaricus, and that's an edible, but there's also poisonous agaricus if you're out collecting agaricus in the woods.
When I first joined IMA, there was a woman who had keyed an Amanita and found it was edible and ate it. But then she realized how close she could have been making a mistake and decided not to even try anymore.
Right. And there's some other edible mushrooms, like some of the parasol mushrooms, especially the Lepidopsina, which is now called Leucoagaricus leucothides, which is kind of mouthful. But that is a nice white parasol mushroom that looks very similar to death angels. It just doesn't have a vulva at the base. So, and then there's also these big parasol mushrooms that come up in the yard, but they have green spores. And that's the most common cause of poisoning because people that don't know what they're doing just see this big mushroom in their yard or their neighbor's yard, and the neighbor comes over and shares you a meal, and half of you get sick and go to the hospital. And we've had several cases of that.
Any more questions?
Question?
I thought I saw somebody back there. Yeah.
First slides, you had some people's RXN.
Oh, reaction,
Ah,
chemical reaction. So RXN is an abbreviation for reaction, so like chemical reactions. And that's sort of an advanced thing if you want to get into more detail on identifying some groups like boletes and some other things. So that's one thing that Michael Kuo especially is good at on his mushroom descriptions. He brings in chemical reactions a lot. So he's one of the few people that test everything with KOH, which is potassium hydroxide, or ammonia. Boletes and stuff, you can also use ferrous sulfate.
From online, how would you describe a light, Laetophorus sulphurus chicken of the wood? How would you describe?
So Laetophorus sulphureus, which that name's going to change, but the American one, the American species turns out different from the US. So look for a publication near you in the future. But Laetophorus sulphureus is a polypore bracket. So it's a shelf mushroom that's in overlapping shelves. It's bright orange on top and bright yellow underneath. It's not gray. It's not white. It's not brown. So ignore half those people on Facebook. The other good edible chicken mushroom is Laetophorus cincinnatus. That is like you take away the yellow. So it's pale orange on top and whitish cream underneath it. The yellow pigment is gone. Any other question? It's named after Cincinnati, Ohio. So that is an American species. So that name will never change.
Variety and doing so much research to the point of I need a dictionary to really look back up on who the adjectives used to describe them. These are so detailed. But within such a detailed process, it seems like there would be room for
Error.
is there just so much debate among mycologists, or is there a general idea of, okay, this is this and that, but I just kind of see this as like there could be a lot of debate real time all the time. I wonder what that may look like, or who could speak to that.
Debate on what the features, how to describe them, or what? Things are shifting. So I'm well, obviously I'm an older guy, but I'm more of a classic taxonomist, collector, alpha taxonomist that goes out and finds them, finds mushrooms, or studies them, and what I used to do a lot with my science research with the Field Museum and previous to that with my graduate work was to write out descriptions, take photos and write out descriptions. Like the cap has this features, the gills do this, the stem has this, the spore color, the veils, what the odor, and then for certain groups, what the taste is. There's a movement online, a movement now to just take a picture and call it done. But you don't a picture doesn't capture everything and doesn't capture all the detail. It doesn't tell you the odor and the taste, for example. It doesn't tell you the chemical reactions. So people like Michael Kuo, Andy Methvin, a whole bunch of people across the country do take time to look at mushroom features. And the more you look at mushrooms, the more differences you find. So this is sort of like bird watching and identifying flowers and trees. You go out and the more you look at the stuff, the more differences you're going to pick up on, especially if you're with other people, to help you notice things. So like beginners on Facebook, they don't know the chicken mushroom has to be orange and yellow. It's not going to be gray or white or brown, things like that. The head of the woods is going to be brown on top and white underneath. It's not going to be gray or staining black or whatever. So It's just like with birds, you look for different shapes and sizes and colors. Mushrooms, the problem with mushrooms, it's a bit with plants, where as the mushroom comes up and ages, the shapes and the colors and stuff can change as the cap opens up and the veil breaks and all that stuff. You get changes through time. So that takes some experience to kind of get familiar with what the mushrooms are doing. But as you said, there's lots of species. So in Chicago area, we have more than 2,000 species of macrofungi. We had a foray on Saturday, and we had a lot of fall mushrooms, not so many boletes and rustlers, but a lot of cortinarius and some tricholoma and some inospes, but a lot of different cortinarius, and we didn't know what most of those cortinarius were, because some of them we hadn't seen before here. They weren't ones I'd seen in Minnesota or Wisconsin. And there's hundreds of cortinarius species. And we had this blue cortinarius, which I had found about 10 years ago. And I couldn't find a name for it, but apparently now somebody has published it, so I have to look that up. But it's an unusual cortinarius because it's a dark blue instead of purple. There's a lot of purple quartz.
Ready for another question? Yeah. Have you discovered any unexpected or category-defining mushrooms that don't fit into their normally described structure? My feeling is with DNA, that's changing all the time.
Well, the thing with DNA sequences and matching up phylogenies and stuff, you find out, you can verify what the species is by matching it up with the DNA library online, which is what we're doing. The people in the club are doing. We're sequencing. They're like taking what we think they are, and after they get a sequence, to see how it matches up this winter or whenever later when they get sequences, and see how close we were or what it is. So we are sequencing a bunch of these cortinarias that we don't know what they are, and hopefully get a match. But one thing that DNA sequencing is telling us is how things are related. So like yesterday, we had 3 different kinds of Flebia group fungi, and they're all in different genera now because they're not all related, but they have similar rubbery, wrinkly undersides, you know. But every year we do find new species, species we haven't seen yet in Chicago or up north. We see new stuff for Wisconsin and stuff. So we also always find unusual things every year.
Could you offer an example where habit is important for identification?
Yeah, so one example, if you're eating mushrooms, would be the elm oyster versus the it's also called the beach mushroom. So there's 2 kinds of Hypsozygus. One grows usually on box elder and elm, and those are big and solid and fat caps with fat stems. And those are usually solitary, which means there's just one sticking out of the knothole. And then the beach mushroom comes up in clumps of 5 or 10 or 20, where all the stems are joined at the base. And that also has water spots on the top, and that's cultivated, and you can get that in the store as a beach mushroom. But that would be a difference in habit. So you've got single versus a clump. And there's differences like that for honey mushrooms and things. Some of them come up out of the ground single, and some are in clumps, for example.
I'm skipping over some things for the moment, but we'll go back.
Any other questions? So the fungal kingdom is less well known than plants and animals. Turns out bacteria are getting fairly well known worldwide. They keep finding the same species, or what they call species, based on sequencing. But fungi, we don't know how many fungi there are in the world, how many wild species there are. There's different estimates ranging from like 3 million to 5 million or 7 million, but we only know 2 to 5%. Only 2 to 5% of those have names. So one thing when you're starting out mushroom identifying, even around here, don't try and identify everything, or don't get discouraged if you can't get a species name on everything. Most things you can find for like a gilled mushroom, you can usually get a genus or group, or you can get that stature type, you know. You can go through, for example, your field guide or whatever you're using, or Michael Quo's website, if you're in Chicago or Midwest, and get down to like what it might be. But Michael Quo's website is excellent, but he doesn't have all the species on there either. You know, he's adding more each year. But there isn't any book or website that has everything, because we don't know everything. And most people starting out in what we call amateurs or non-professionals, they don't really have access to the technical literature. I do, but I don't have the time to spend hours and hours under the microscope looking at all of it. I don't enjoy that. So I tend not to identify mycena, and I have a colleague that does russulas and boletes and some other stuff. But yeah, that's the problem with mushrooms and fungi, is we have so many that don't have names. But in the macrofungi group, we probably, I don't know if anybody does an estimate, but we probably have a higher percentage of species identified or named under macrofungi versus microfungi. Microfungi are microscopic fungi you're not going to see unless you culture them out of the soil or water. So, and a lot of plant parasites and animal parasites are unknown.
Any suggestions on where to see different stages of a mushroom, for example?
Out in the woods, you can see. So,
I mean, you got nice pictures of that stuff.
right. So that's what, so some mushrooms will come up a bunch at once, but some, like an amanita, might just come up one, and it's going to be at whatever stage you find it. And one thing I keep reminding myself, some of these non-mushroom types, it might not be obvious that it's still young. So like there's these Celefera and ground fans and subsina type things. They come out of the ground and they grow up. And if it's young, it's going to be short and stubby. And you may not realize that it's not fully grown. Because if it's a small mushroom, you would have the little cap and stem, you know, that hasn't opened up yet. But yeah, so if you're lucky, you'll see different stages of the same mushroom near each other. But it just kind of go out and look in your yard or neighborhood or out the woods.
Do mushrooms hybridize such that it's sometimes impossible to tell what you've got?
So there's very few cases we know of mushrooms hybridizing. So they don't hybridize a lot like plants do. So plants, you can hybridize those by moving pollen around between flowers and stuff. And some plants don't rely on the pollinators to distinguish the different flowers rather than identify whether it's the same species or not. Fungi have mating genes, so that when the hyphae come together, they recognize if they're the same species and they have different mating type. So I don't know really, I don't remember any cases where we have hybrid mushroom species.
That's okay.
We do have different varieties or forms. So we have species, and then those species will have different variation across their geographic range in their size and kind of shapes and colors. And like 100 years ago, there were more variety names and form names. So form would be like a different color or whatever. One feature is different in a form. Two or more features are different in a variety. And we don't use those levels under a species name quite as much as we do anymore. So there is variation across the country or world for different species.
So this is a question I think rather broad, but what about mushrooms that glow in the dark, bioluminesce? Now, we do have that program from what, a year and a half, 2 years ago, all on that topic
Oh, yeah, you mean,
page.
yeah. So we have several different mushrooms that are bioluminescent, and they're not necessarily related. So for Chicago area, we have the jack-o'-lantern mushroom, which is the big mushroom that's orange, and the gills glow green. We have the Pinella stypticus, which is a small little mushroom with a stubby stem on wood, and that can, if it's fresh, that can glow really bright green. And then we have the Armillaria mellia in that group with the rhizomorphs and cords. And that's called firefox or foxfire, sorry, that's foxfire. And that is the mycelium in the wood glows, and I've never seen that. But if you go out at night and you tear open a rotted log, you might have a chance of finding that. But people would see that down on timbers in the cave or timbers in a mine and stuff like that, where the armillaria is rotting the wood. They'd see this what's called foxfire.
Well, I'll ask this question. Is there a hen of the woods, and is that different from chicken of the woods?
Yes.
Yes.
So there's a whole bunch of poultry of the woods. You want to call them that? So the long standing common names are chicken mushroom, which you can also call sulfur shells. That's orange and yellow. There's hen of the woods. And when I write out hen of the woods, I put hyphens in there, just like the old man of the woods. I would put hyphens, old hyphen man, hyphen of, hyphen the, hyphen woods. Because it's not an old man in the woods, it's the old man of the woods. That's one common name. So hen of the woods, and that's Graphola frondosa. So it's gray on top and white underneath, and it's in roughly overlapping lobes like a hen, ruffled hen. So we have these two names. So somebody, I don't remember who. So there's a movement to give everything a common name, and iNaturalist is one of those sources that is generating common names. So now we have rooster of the woods, and you can look up and see if you can figure out what that is. And then there's another mushroom which had a perfectly good name called fried chicken mushroom that is now called by one person that edited a Wikipedia page. And he calls it, what does he call it? Chicken in the road or something like that. Because he finds it in the trails or paths.
And he's the expert.
And I haven't seen anybody else. So there's all sorts of weird names that you can find.
I have a final question for
Okay.
you that falls into this category. Well,
Okay.
I'll let you make the judgment. I heard mushrooms are more closely related to humans than they are plants. Is this true? And if so, why?
Yes. So I follow a bunch of YouTube channels on YouTube. And there's a science channel that had, so these YouTube videos have to have catchy clickbait titles. So there was one a couple weeks ago that said, "You are basically a mushroom." And I was like, no, we're not. So I watched the video, and they actually didn't say that in the video. They pointed out how fungi and animals are related. So the fungus kingdom shares an ancestor with I have a whole nother talk on this, but which is you can find online, Fungi and the Tree of Life. But we can have it again. I think they've done that one.
Yeah.
But so the fungus kingdom shares an ancestor with the animal kingdom. And that pair of animal fungus lineage shares an ancestor with the amoebozoa, and that has the slime molds and the amoeba type stuff. And that's a separate lineage from all the other eukaryotes. So the plants are all way in a different part of the tree with green algae and red algae and some stuff. So there's no relationship between plants and mushrooms. But because mushrooms don't run around and eat things, they were put in plant kingdom way back. And the fungus kingdom wasn't actually its own kingdom until like the 1950s, which is pretty recent. And at that time, it became the fifth kingdom. So you might see that.
This was great.
Thank you.
I'm so glad you spent your time with us today.
Thank you.
Thanks so much, Patrick.
See ya.